Next, we will hear from Saoussane Khalifa at Tohoku University in Japan. She will be presenting on exposure of squalene hydroperoxides to photons causes the formation of unique squalene cyclic peroxides by a chain reaction mechanism detected on the human skin and causing harm to skin cells. Saoussane?
Yes, so thank you very much for introducing me, and thank you everyone for joining in. So without further ado, I will start my presentation about squalene novel cyclic peroxides, their detection on the human skin, and their effect on skin cells. So to begin with, our skin is the first barrier that separates us and that protects us from the external environment, and our skin is coated with a layer of lipids that plays an overall protective role. However, when these lipids are exposed to sunlight and to other environmental stressors, they undergo oxidative modifications to form harmful oxidation products. So if we look at the composition of these lipid species on our skin, we will find that squalene accounts for 12% of total skin surface lipids, so which makes it the most abundant skin surface lipid. And it has been already demonstrated that when our skin is exposed to photons, for example, sunlight, the photosensitizers that we have on our skin, they transform the triplet state molecular oxygen, which is the oxygen found in the air, to singlet oxygen, which is a very reactive form of oxygen. So when this singlet oxygen is formed, it reacts with squalene and it attacks the double bonds present on the molecule. So when it attacks the double bonds, it forms squalene monohydroperoxide isomers in the number of 6 isomers. So of course, the difference between the isomers is the position of the hydroperoxide group on the molecule. So these hydroperoxides are relatively unstable because of the presence of the hydroperoxide group. And since our skin is constantly exposed to sunlight and to other environmental stressors, and since SQOH, which are squalene monohydroperoxide isomers, are relatively unstable, we assumed that these squalene monohydroperoxide isomers can actually form other products when they are further exposed to photons and in the presence of triplet state molecular oxygen, which is the oxygen found in the air. So what we did next is that we synthesized pure SQOH isomers and we exposed each isomer individually to photons in the presence of triplet state molecular oxygen. The results that we found were very interesting because the mechanism that was formed, the mechanism that enabled the transformation of these SQOH isomers proceeded by a chain reaction mechanism, and it had a pattern which was compatible between each SQOH isomer. So among the cyclic peroxide species that we found, the two species 2OH3,1,2-dioxane SQ and 2OH3,7-di-1,2-dioxane SQ, which are the only two cyclic peroxide species presented here, were actually detected on the human skin. Not only that, but 2OH3,1,2-dioxane SQ, which is highlighted in a blue frame here, was actually detected inside the epidermis in the top layer, which is the stratum corneum layer. So since we saw that this compound was detected on the surface and it can penetrate the epidermis, we wanted to check its effect on skin cells. So we used keratinocytes, which are cells that make up 90% of the epidermis, and we treated these cells with 2OH3,1,2-dioxane SQ. So when we cultured these cells, we cultured both the basal phenotype of the cells and the fully differentiated cells, which mimics the epidermal model of human skin. So when we treated these cells with 2OH3,1,2-dioxane SQ, we saw that there was an upregulation of keratin-1 gene, which is responsible for differentiation and keratinization. The upregulation of interleukin-8 and interleukin-1 beta was also observed, which are cytokines that cause inflammation. There was also an upregulation in the COX-2 gene, which is also related to inflammation. So when we used higher doses of 2OH3,1,2-dioxane SQ, we observed a significant decrease in the viability of the cells, which means cell death. So if we look at all these symptoms, accelerated differentiation, keratinization, and induction of inflammation, also cell death, these are symptoms often observed in subjects with altered skin conditions, such as psoriasis and atopic dermatitis. So these compounds, we didn't even know that we had them on our skin, and they are causing significant harm to our skin cells. So subjects with skin disease might have elevated concentrations of these species. It is also necessary to scan for similar compounds that can be formed from other lipid species on our skin and to find a way to stop the mechanism leading to their formation. So this is all for my presentation. Thank you very much for your attention. And if you have any questions, please don't hesitate to ask.
Thank you, Saoussane. That looks like a very complicated and difficult set of reactions to carry out. It is, yes.
Yeah.
So thank you. I have a question here about the keratinocytes you tested with.
Yes.
So did you use cell lines that you have cultured and kept in Petri dishes, or what kind of cells did you have for that?
Actually, we used HaCaT cells. So HaCaT cells is an immortalized cell line. The reason why we use this type of cell line because it can maintain the same expression of genes over a long period of time. Whereas if we use normal cells, they have a very limited lifespan. It's about 3 weeks and the cells die. And the turnover of gene expression during that period is very, very different. So HaCaT cells mimic the human living cells the best. And also because we needed to do differentiation, it's better to do it on HaCaT cells.
So you maintain the cell line in the undifferentiated state, and then you differentiated it. Okay.
Exactly, yeah. So we used higher calcium levels. We maintained calcium levels at a low concentration to maintain the basal phenotype. And we increased the concentration of calcium gradually to obtain the fully differentiated keratinocytes.
Interesting. I believe Japan is very advanced in this kind of skin research.
Yes, actually it is.
Yeah.
Yeah, and this work actually is very hard and long. It's the work of 3 years. This is all, yeah, because the identification of the compounds takes a lot of time, the synthesis and the, yeah.
Yeah, just making those compounds had to be a challenge.
Yes, it was very challenging. And the identification was the most challenging part, actually.
Really?
Yeah, because we had to use NMR, derivatization reactions, and even chemical calculations using Spartan AT software. So it was very hard, yeah.
That's great. Congratulations.
Thank you.
Do our judges have any questions?
I can start off this time. So Saoussane, thank you for the very nice presentation. You made me wanting to put more sunscreen on my skin.
Yes, we should.
So I'm curious about the application implication of this study. What do you think would be a solution to slow down this type of reaction except putting more sunscreen?
Actually, I think instead of putting more sunscreen, I think eating food that has a lot of antioxidants will be better. Maintaining a more healthy lifestyle, because there are a lot of antioxidants like vitamin E that are that act mainly on the skin. Because, yeah, so I think maintaining a healthy lifestyle is better than putting on a lot of products.
Okay, thank you.
Yes, you're welcome.
So I can follow up with one more question. I actually had the same question as Nora, but when you studied these reactions, if I understood it correctly, where you had the original squalene peroxides, and then you did the reactions, but in which system did you do those reactions?
So first of all, to produce squalene monohydroperoxides, I oxidized squalene using singlet oxygen. So to produce singlet oxygen, I had to use a photosensitizer. I used rose bengal. So rose bengal is a type 2 photosensitizer. When it's excited with light, it produces singlet oxygen from triplet state molecular oxygen. So by singlet oxygen, I produced squalene monohydroperoxide isomers, and then I purified the isomers using HPLC in normal and reverse phase. And then once I obtained the pure compounds, I oxidized each isomer alone, and I saw the compounds that could be formed.
But what was the system that you did the oxidation in? I mean, was it in a solvent? Was it in an oil, or how did you do that?
Yeah, so the first oxidation to produce squalene monohydroperoxides, I oxidized in ethanol because it needs to proceed in protic solvents. And the second oxidation to produce cyclic peroxides, actually it proceeds only in aprotic solvents or in a medium with no solvents, in a system with no solvent.
Okay.
So if you expose SQOH with light and triplet state molecular oxygen, you will get cyclic peroxides.
Okay, thank you.
No problem, thank you.
So then I'll kind of trail on that question is, I imagine some of these are fairly unstable. How do you stabilize them enough to be able to analyze them?
Actually, that's surprising because cyclic peroxides, I thought that they will be more unstable, but actually they were more stable than hydroperoxides. The 4-membered cyclic peroxides are more unstable. So this one is a dioxine. It's a 6-membered cyclic peroxide. And I found that it's relatively more stable than hydroperoxides. When I tried even to oxidize it more, it was more resistant.
Wow.
So for hydroperoxides, I maintain them in protic solvents. Because if there is a chance of deprotonation, there is always a source of protons. So I maintain them at -80 degrees in protic solvents, and they should be fine.
Thank you.
No problem.